We are now going to discuss cell crawling. Cell crawling allows the cell to cross a given surface. We're going to discuss cell crawling, actin polymerization, types of actin, and mechanisms. Cell crawling uses actin filaments that create a mesh that exerts the membrane outwards. This mesh is known as lamellipodia. Actin is then flattened and extended through thin projections outwards. These thin projections are known as filopodia. Actin is an accessory protein, holding actin into a mesh and controlling these proteins in order to use a master actin controller. This master actin controller is known as Rho. Rho is a bundle of two solid tubes that allows for the regulation of actin. The cells must anchor with integrins and bind to the actin inside of the cell. The cell will then drag itself forward as actin coats the inner surface of the cell, which is known as the cell cortex. This cell cortex binds to actin and uses myosin motors to pull the cell forward. When the cell anchors, it uses transmembrane proteins and their function by signaling sequences to go to the ER using stop sequences to transfer those proteins and embed them into the surface. Here you can see the cell crawling and extending those lamellipodia and filopodia extensions, actin extensions.